This is a continuation of Chapter 5. This is Section 5F. So our opening question for this section is, can we predict inheritance patterns of genes? And for example, for some people, the chemical PTC tastes very, very bitter. And yet others, it's tasteless. We actually used to do this experiment in the lab where we had paper that actually had PTC on it, and people would let it, touch it to their tongues. And it was always interesting. You could always tell if people were a taster or a non-taster, because the tasters, they would put, the paper with the chemical on their tongue, and they would like grimace. They'd be like, whoa, it tastes terrible. And then the people who were non-tasters, they would put the paper on their tongue with the chemical, and they'd look at you like, so what's the big deal here? I don't taste anything. And it turns out that this is genetically controlled. Scientists report that the ability to taste PTC is generally a dominant trait. So because it is a dominant trait, it is represented by a capital T. And the non-taster is represented by a lowercase t. So what is the genotype of a non-taster? Well, a non-taster, to have the recessive trait, the phenotype, to have the recessive phenotype, the only way to have that is to have two lower t's. Because if you had a capital T, the capital T would sort of overshadow the small t. So what is the genotype of a non-taster? It would be two lowercase t's. And I'm sorry, I know I'm not very good at writing this. Okay, could a non-taster's parents be tasters? What do you think about that? Is it possible that this non-taster, could they have parents that were tasters? So think about that. But remember, if you are a taster, remember you have to have two alleles, two letters for every trait, because you get one from your mom, one from your dad. And remember that if you have a capital T, which is a taster, you could have two possibilities. If you are a taster, you could either be capital T, or you could be capital T, small t. All right? Now, both of these, their phenotype would be, they would be tasters. They'd be one of those ones to put it on their tongue and be like, oh, this tastes terrible, okay? All right? And you wouldn't, by just looking at them, you would not be able to tell the difference between this. Remember, this is homozygous, and this is heterozygous, okay? Homozygous means the two alleles are the same. Heterozygous means they are different. Okay, so looking at them, you wouldn't be able to tell, all right, whether they're homozygous or heterozygous. But the question back to this is it possible for two tasters to actually have a child that is a non-taster? And the answer is yes. If both parents had this genotype with the capital T and the small t, you would have, so one parent could be this, capital T, small t, and the other parent could be this, capital T, small t. And when they made their gametes, they would either have capital T or small t, okay? And it is possible that two of the gametes that were little t, lower t, could come together in fertilization and give you a non-taster. Okay, so two tasters could have a non-taster child. Now, I kind of did it messy there, and there's actually a neater way to do this. And that's using a Punnett square. So a Punnett square can be used to predict the results of a genetic cross. And in a genetic cross, you have two parents, and that's called the P generation. And they are crossed to form offspring, and that's the F1 generation. So you have the P generation, the F1 generation, and then you can actually have an F2 generation. The Punnett squares are named after a British geneticist named Reginald Punnett. And they are very, very useful. Here's just more adorable black labs, chocolate labs, and showing you that coat color is genetic, as well as deafness actually in these dogs. So these dogs can also be deaf, and that is genetically controlled as well. So let's do what's called a simple monohybrid cross. A cross between two individuals that are that you're dealing with just one character is called a monohybrid cross. So here represents, here's one parent. Okay, this is the male parent, and here's the female parent. Okay, because you can tell this because these are the eggs right here, and these are the sperm. Okay? Now, in this particular cross, you have, remember, capital B is going to be black, and little b is going to be chocolate. Okay? And remember, for every trait, you're going to have two letters represented. And then the question is, are they two letters that are the same or two letters that are different? Okay. So in this case, the mom is homozygous dominant. Now, the reason I say that is homozygous because both letters are the same, and dominant because both letters are capital. So they're capital and they're the same. So the mom was homozygous dominant. The dad, which you can see from the sperm up here, are these the same or different? Well, hopefully you said they're the same. So the dad was homozygous also, but are these letters lowercase or uppercase? Well, hopefully you said lowercase. So because they are lowercase, the dad was homozygous recessive. So one of the ideas here is that when, so the mom was capital B, capital B. So here's the mom. Now when she makes her eggs, remember these two alleles, because that's what these are, these two alleles will separate. So this mom can make two types of alleles, and that's what you see here. Well, actually, she can only make one type of allele, and they're all capital B. And the dad, let me do a different color here. So here's the dad. The dad would be homozygous recessive, so he's going to be two little b's. Now, when he makes his sperm, which would be like this, okay, that's what's up here. The only thing that he can give them is a lowercase b. So the thing about a Punnett square is it represents fertilization events, and it's a really easy way to keep track of the different possibilities for fertilization events. So do you see how this black lab right here is the result of this egg combining with this sperm? So you have a capital B, which is right here, and a lowercase b. And that's what you see here, a capital B, little b. Okay? Now remember, the capital B is black and the little b is chocolate. And remember, the black one, because it is dominant, will overshadow the recessive. So when you have a capital B and a small b, this puppy will be heterozygous, okay, one of each type, but that's their genotype will be capital B, little b. They will be heterozygous, but their phenotype, their observable trait, is they will be a black lab. And in fact, all of these puppies have the same combination of genes, big B, little b. So they'll all be black labs, and they'll all be heterozygous. Now, let's say you take a heterozygous, the last one was homozygous. Let's say you take a heterozygous mom. Now she will look like a black lab, all right? She will, but her genotype will be capital B, small b. And let's say you cross that mom with a dad. Here's the dad right here. And the dad is still homozygous recessive. So he's a chocolate lab, you can see. So he's small b, small b. Now you have a new combination. Do you see how in this case all the puppies are not the same? So if you look at this square, that's the combination of this big B from the mom and this little b from the dad. So big B, little b, but remember the big B is going to overshadow the little b, so the genotype will be big B, little b, but the phenotype will be black. And that's the same that happens here. But now let's look down here. Here you have, I'll do a different color. Here you have the little b from the mom, and you have a little b from the dad. So then you get a combination of little b, little b. Little b, little b. Remember, the small b is the chocolate trait. And so when the two little b's get together, you have a chocolate lab. And there is that little adorable chocolate lab. Okay? And the same thing here. You have a small b and a small b. And so you have little b, and it's going to be a chocolate lab. Okay? So you should be able to do this using the Punnett squares. And you will get more examples of genetic problems in lab, but you absolutely will have them on the exam in lecture as well. Okay? All right, so let's go back to thinking about meiosis a bit. Remember, with meiosis, alleles separate, right? So alleles, right, which are two like variations of a gene, will separate during meiosis because remember, homologous pairs separate during meiosis. Now, Mendel had a couple laws that he developed regarding genetics, and one of them is called the law of segregation. And what the law of segregation just says is that the two alleles for a character will separate during gamete formation. So remember, gametes are egg and sperm, and they exist when you undergo meiosis. And remember, here is a homologous pair, and during meiosis, homologous pairs separate. And so if you have a heterozygous black lab, like this one here, this mom, she will have a big B and a small b for her genotype. But when she makes her eggs, one allele will go in one type of egg, and the other allele will go in the other type of egg. So during meiosis, you have this segregation. The two alleles separate, and they go in separate gametes. So this mom can actually make eggs that are have a capital B. 50% of them will have a capital B, and 50% of them will have a small b. And what ends up with the puppies just depends on which bee that particular fertilization event ends up with. So I mentioned to you before that when you see an individual like this black lab that has the dominant trait, you don't really know their genotype for sure. You know they have one capital B because they have a black coat, but you don't know about this other allele, because remember, to have a black coat, you could either have a capital B, or you could have a capital B, small b. All right. Now there is a way to test for this, and it's called a test cross. And the way you do this is if you do a test cross with a homozygous recessive, in this case it's the chocolate lab, little b, and then you look at like a litter of puppies in this case, if a chocolate lab comes up with the puppies, then you know that the mom was heterozygous, because remember, the only way to get this chocolate lab is to have a little b, capital B, little b. So here is the test cross. What would happen if the dog was homozygous dominant? So she would be capital B, capital B. You do the test cross with a homozygous recessive, little b. When you actually do this, you can see that all four puppies combinations are big B, little b. So all of these guys will be black labs. So if you have, I don't know if you've ever, known a dog breeder or something, but if you were to have a black mom, a black lab mom or dad that's, and you cross them with a chocolate lab, and you did it several times, and they had a bunch of puppies, and they were all black. They were all black labs. Okay, then you could be fairly confident that, in this case, it's the mom, because here's the egg. You can be fairly confident if you only see black lab puppies, you can be pretty confident that the mom was homozygous dominant. Now, on the other hand, if you do this experiment with the dad being homozygous recessive, the mom turns out to be, you're not sure, but if you do, the mom is a black dog, and if she is heterozygous, big B, little b, then 50% of the puppies, there's a 50% chance of having chocolate labs. Now, that doesn't mean that in real life, that the mom and the dad dog have 4 puppies, and 2 will always be black labs and 2 will always be chocolate labs. But in a sense of probability, probability states that you have a 50% chance of having a chocolate lab puppy and a 50% chance of having a black lab puppy. But back to the question, how do you know whether the mom here, who is a black lab, is homozygous or heterozygous? If you do this test cross and you see chocolate puppies, then you know that the mom was heterozygous. Now, the next thing we're going to talk about, everything we've talked about right now has been looking at one single trait, coat color, okay? That's called a monohybrid cross. You're looking at one trait with two different alleles, or later we might look for multiple alleles, okay? But the idea is, can we actually look at more than one trait at one time and do predictions? So, for example, Labrador retrievers, breeders need to keep track of the traits for coat color and hearing because there is this possibility of the dogs being deaf. There's a genetic trait of deafness. So the question is if you wanted all chocolate coats, how would you avoid breeding deaf puppies? So you would have to keep track of both the deafness gene and the coat color gene. And it is possible to do that because what happens is, remember, when you have meiosis occur, the alleles are going to separate independently during gamete formation. And this is called the law of independent assortment. This is another law that Mendel came up with. It states that the inheritance of one character has no effect on the inheritance of another character. They sort independently of one another. So the character of deafness versus the character of coat color, you can figure out all the different combinations. They will be independent of one another. And this is what that actually looks like. So remember, we talked about this idea of independent assortment. Okay? Independent assortment is that each pair of alleles, either big B, little b, or big D, little d, will separate individually from the other pair of alleles. So that, and when they come back together during fertilization, these are all fertilization events, you will get these different alleles, okay? So this is actually a little boy dog. This is with a black coat, okay? And this dog is also can hear. It's not a deaf dog, okay? However, for both coat color and deafness, this male dog is heterozygous. You can tell because it has a big B and a little b, and a big D and a little d. Now, when this dog makes his sperm during meiosis, each gamete, each sperm must have one of each letter. Do you see how this one has a big B and a big D? This one has a small b and a small d, big B, big D, and then this has a big B, little d. This has a little b, big D, okay? And these different gametes, these different sperm, result from the idea that when this dog makes the sperm, this chromosome, okay, with this set of alleles will go independently from this chromosome with this set of alleles. And so the only rule is you have to have one of each letter in the sperm. So this big B can combine with the big D, okay? And that's what you have over here, okay? Or you can have this big B combining with the little d. And when you have that, then you have this combination, okay? Or you can have this little b, which you can't see because I've already like messed everything up, but that's a little b, can combine with the big D, okay? And that's what you have here, the little b and the big D. Or in the last combination is you can have a little b and a little d, okay? And that's the combination you have right here. All right, little b, little d. You can actually, if you're trying to think of all the combinations, you can actually do kind of a pseudo Punnett square. If you're trying to figure out, okay, what do the gametes look like? And you can put like a capital D here and a little d here and a capital B here and a little b here. And actually, if you do this, you know, you'll have big B, big D here. That's this one. You'll have a big B, little d here, and that's this one. You'll have a little b, big D here. That's this one. And you'll have a little b, little d here. That's this one. Okay? So the general idea is that when you make gametes with two different traits, they will sort independently and you will have all the different possible combinations. Now, let's talk a little bit about, you can do what's called a dihybrid cross, okay, where you can keep track of two different traits. Okay, so in this case, you have a male that has a black coat and is hearing and is heterozygous for both traits. And the same thing with the mom. And you can do like a mega Punnett square, which is 4 by 4 with 16 squares. And you can get the data. And typically when you do this, it is in what's called a 9:3 to 3:1 ratio, where the 9 are the two dominant traits, the 1 are the two recessive traits, and the 3s are one dominant, one recessive trait. Okay? And this is called a dihybrid cross. So we're going to start to talk about understanding inheritance and using that to be able to predict the occurrence of certain genetic diseases. And our case study is with Dave and Katie. They are healthy adults, but both of them have a sibling with cystic fibrosis. And cystic fibrosis is a very, very serious disease, a life-threatening disease that is an autosomal recessive genetic disorder. Wow, that sounds like a mouthful, doesn't it? But what it is you have a autosomal. Autosomal means that it is on one of the autosomal chromosomes, one of the autosomes, not a sex chromosome. It is a recessive disorder, meaning that for the disease to take place, you must have two recessive alleles, which would be represented as two lowercase letters. And it is a genetic disorder, meaning that it is inherited. So Katie and Dave underwent genetic testing, and they found out that they were both heterozygous. So what you're supposed to do is imagine you are a genetic counselor and explain to them their probability of having a child with cystic fibrosis and show your work. Okay, so what you need to do is you need to set up a Punnett square and then figure out, knowing that Katie and Dave are both heterozygous, what is the probability that they could have a child with the recessive cystic fibrosis. Okay, so the first thing you want to do is decide on letters to represent cystic fibrosis. Okay, they could be any letters. I think in the book they often use A for normal, and capital A for normal and small a for the disease if it's a recessive allele disease. But a lot of times I pick letters that have to do somehow with the actual disease, just because it's a little bit easier to know. But you want to pick a letter that the capital and the lowercase look different. It just makes it a little easier. So C's are not good because a capital C and a lower C kind of looks the same. So I actually would suggest using F, okay, where capital F is normal and small f is the disease allele. And it tells you that Katie and Dave are heterozygous. Remember, hetero means different. So what would their genotype be? Think about that. You need two letters. Why don't you use F's? Are you going to have two capital F's, two little f's, or are you going to have one capital and one lowercase f? And so if you can get to that and then set up a Punnett square, that would be great. If you can't, so if you can, stop the recording and draw out your Punnett square. If you're not sure how to do it, get yourself a piece of paper and a pencil, and I'm going to walk you through it. Okay? So we're going to start by drawing out a key where capital F would equal normal, the normal allele, no disease, and the lowercase f would be the cystic fibrosis disease allele. Okay? Then the next thing I usually do with a genetic type of question is I draw what's called a pedigree. And we'll be talking about pedigrees in a few minutes, but why don't we just start now? So a pedigree is kind of like a family tree. So, if some people study genealogy, they will have these pedigree charts. But you can also have them to keep track of genetic traits. So in a pedigree chart, the circle is represents a female, and the square represents a male. Okay, a diamond actually represents that you're not sure what the sex of the child is because it's not born yet. Okay, when you have this horizontal line between a circle and a square, that represents a union. Okay, so this is like the mom on the left in the circle, and this is the dad on the right with the square. And then when you have a line going down from that horizontal, that usually indicates a child. Okay, and if they can have more than one child, and then you kind of have sort of you can see more shapes as their children. So, but we're just going to go with this, with the diamond representing this unborn child. If they were to have a child, what is the chance that that child would have cystic fibrosis? So we already talked about that Katie and Dave are both heterozygous. So that's why I put capital F and little f here, capital F and little f here. Now the next thing you need to do is you need to set up the Punnett square. And I have the Punnett square over here. Okay, so if you know how to set up the Punnett square, if you've go ahead and do that. Okay, if you haven't yet, let me give you a couple little hints. So I usually, it doesn't matter, but I usually put the mom along the side here. And I usually put the dad along the top here, but it really doesn't matter, but it just is like if you get a system, it's just easier. Now remember, the mom is heterozygous. When she makes her eggs, she can make 2 different types of eggs. Now, what are the 2 different types of eggs she can make? Remember, Mendel would say that when gametes are made, that these alleles separate. So half the gametes will be a capital F, and half the gametes will be a small f. So you can put a capital F here, and you can put a small f here. And then the next thing that you want to do is do the same thing for the dad. Well, the dad is also heterozygous, so when he makes his sperm, same thing. Half the sperm will have a capital F allele, and half the sperm will have a small f allele. So that you can draw that here, capital F. I hope you're like neater than I am. And here's a small f. All right. Now the next thing you need to do is you need to put the letters in each box, right? And remember, each box, each rectangle represents a fertilization event. So this in here, right, you would have big F, big F. Okay? All right, so go ahead and finish that. Just stop the recording and finish up your Punnett square. Okay? And then start to think about what those are the genotypes. What would the phenotypes be? Would in each box, would that be a normal child, or would that be a child with a disease? Okay? I will say there's something else, and that's called a carrier. Now, when a person is heterozygous, they will appear normal, and that's the case of Katie and Dave. They don't have the disease, but they are what's called carriers because they are heterozygous, and so they are carrying the cystic fibrosis allele, but remember it's hidden because their other allele is a normal allele. So go ahead and finish up your Punnett square. And so you're going to hopefully finish it, and you will have gotten an F, F, and you will have an capital F, small f, and you'll have another capital. You tend to put the uppercase first, so capital F, small f, and then small f, small f. All right, so hopefully you got that far. So let's begin to analyze it and look at, this is the genotype, let's talk about the phenotype. So with the phenotype, remember the phenotype is the observable trait. So of these 4 blocks, do any of these 4 blocks have a phenotype that would be cystic fibrosis? Because remember, the question you're trying to ask is, you know, is it possible for this couple to have a child with cystic fibrosis? And what do you think? Well, the answer is yes, because look at this small lowercase f, lowercase f. This is the genotype for cystic fibrosis, 2 lowercase disease alleles. The rest of these, this one, and this one, these guys will all appear normal. They will be, their phenotype will be normal. Now, 2 out of the 3 will be carriers, right? This one and this one. So their child could be a carrier. They could not carry it at all, or they could actually have the disease. And a lot of times we express this, the phenotype and the genotype, as either ratios or percentages. Now, I tend to go with percentages because I kind of think about, you know, just like a pie chart or something like that. So in this case, 75%, there's a 75% chance that the child will be normal, although there's a 50% chance that the child will be a carrier. And there's a 25% chance that the child will have cystic fibrosis. Okay? Now, the thing to remember about this probability is, let's say Katie and Dave decide, okay, we're gonna try to have a child, even though there's a 25% chance of having cystic fibrosis. Okay, and they have a child, and the child has cystic fibrosis. Okay, so then they decide to have a second child. The question is, what is the probability that their second child will have cystic fibrosis? Any ideas? Well, it's actually 25% again. Okay, it's 25% again. This represents, during each fertilization event, the probability of having one type of genotype over another. It's kind of like when you have a, you know, if you want a boy or a girl, you know, if you have, you know, you have a 50%, roughly 50% chance of having a boy or a girl. So if the first time you have a child, you have a girl, that doesn't mean that the second time you have a child, you're going to have a boy, right? The second time you have a child, you have a 50% chance of having a boy or a girl again. Okay? So we want to talk a little bit more about sort of genetic characters and how genes control genetic characters. So here's probably 2 siblings. There's the freckled phenotype, which is dominant over the non-freckled phenotype. And talking about this idea of human genetic disorders, many are recessive. Some are dominant, but many are recessive. A carrier is a heterozygous individual. Carriers do not have the disease, but they can pass it on to offspring. And this is basically what we just talked about with the cystic fibrosis, right? Where you have 2 parents that are heterozygous, and if it is a recessive trait, there will be, if both parents are heterozygous, there will be a 25% chance of having a child with that particular trait. So here's a fancier pedigree where you can see that so here is the first generation, the second generation, and the third generation. Here you have 2 parents that are heterozygous. Okay, that's grandma and grandpa. And for each fertilization event, each child, they have a 25% chance of having a child with the trait. And here's an example of the child with the trait. This person marries, so this is not a brother. This guy's not a brother. This is a husband. Okay, she marries this guy. He is heterozygous. She is homozygous recessive, right? And when they have children, this child, I mean, in yellow, that is recognized, they are recognizing that as a person with the trait. So they have 1 child with the trait, and then they actually have 3 children that are heterozygous, that are carriers.